Hi guys, my name is Andrew Molnar and I'm here to talk to you about the Human Genome Project that I worked on with Andrew Stansfield, Jared Peters, and Luke Lefevre. So to give a little background about the Human Genome Project, we're going to dive right into the technology behind it. So a little overview of the project itself is that the Human Genome Project was a 13-year-long publicly funded project starting in 1990 with the goal of determining the DNA sequence of the entire Eurochromatic human genome in 15 years. In the beginning, the Human Genome Project was criticized by many people, scientists and non-scientists. One big question was whether the huge cost of the project would outweigh the potential benefits, which is still being determined. So diving into the technology behind it a little bit more, we look at 3 steps. Step 1 is to obtain a DNA clone to sequence. As you can see from the picture, we get the nucleus from the cell, the chromosome from the nucleus, and the DNA from the chromosome. Step 2 is to sequence the DNA clone. This is done by pairing A and T and G and C. Step 3 is to assemble sequence data from multiple clones to determine overlap and establish a continuous sequence. This video shows a little bit more about the overview of the Human Genome Project.
Thanks for stopping by. Today we are talking about the Human Genome Project. Before we discuss the actual project, I want to define what a genome is. So a genome is the complete set of genes in an organism. In the Human Genome Project, that organism is, of course, the human. There are several scientific techniques that made it possible to start and complete the Human Genome Project. These include DNA sequencing, polymerase chain reaction, and electrophoresis. The Human Genome Project was essentially an international research project to map the entire human genome, every section of DNA in a human cell, which is a lot, 3 billion base pairs, in fact, and they've mapped all of them, which took 13 years to complete. The project determined that there are about 20,500 genes in the human genome, and it also discovered the order and location of these genes. It didn't, however, identify what each gene does, and that research is ongoing and there's still a lot to learn. It continues to be a benefit to society as a massive information source for researchers to draw on and to continue their advances in both science and medicine.
With new technology and the ability to test genomic data, everyday people will have access to their genome information. Ethical decisions come into play regarding the release of this genome information. There are debates regarding whether genome test results should be released to those who agree or sign up to be tested. First of all, results do not prove conditions. You can find a genomic pattern for a different condition than you were tested for, and there is more questions whether results should be public for better testing and input. Genetic makeup does not guarantee a future condition. If your DNA shows the likelihood of developing Alzheimer's when you're older, it is not certain. There are external factors that can impact that diagnosis in the future. As a doctor or analyst, are you responsible for giving them their results, whether they are positive or negative? There are potential legal issues if the results aren't released. Let's say someone comes to get tested for the genetic makeup of a heart condition, but you recognize a gene that leads to a brain condition. These results are contradicting to what the person was tested for. Are you responsible for telling them what you discovered regarding the brain and the heart? If this person dies from a brain condition that you realize from their genetics, but you don't tell them, are you responsible for their death? With all this new genome data being collected, are scientists ethically obligated to release data and information to help future studies, or is this sensitive information that needs to remain private? The biggest concern regarding telling participants about their genetic makeup is the potential change to their lifestyle when they found out their results. Some people who find that they have a life-threatening condition in their DNA will choose to live life to the fullest. They will travel the world or go skydiving. Others could gain anxiety regarding their condition and seclude themselves from society. Either way, their lifestyle will change. A Slate study regarding a genetic test for Huntington's disease showed that roughly 70% of those at risk said they could take a test if it existed, and they would take the test. In fact, only 15% do. People don't want to be tested or given results because they know that it will change their mindset and their lifestyle.
A rather extreme possibility of the Human Genome Project is designer babies. Designer babies are essentially just babies that are modified, presumably by their parents, to have more desirable qualities or to not have qualities that are undesired, like genetic diseases. Now, to many, this is humanity's next step in its evolution. After all, there are many benefits, such as the child and the individual being significantly healthier, being more physically fit and more inclined to be physically fit, being more intelligent, or in some ways saying making more money, and, of course, other more cosmetic benefits, such as never being bald, having the eye color that you desire, etc. Now, though there are many benefits, there are, of course, extreme risks involved. After all, if you decide that you don't want a designer baby, then you will be accepting the fact that your kid will have to compete in a world with other designer babies who are incredibly intelligent, incredibly athletic, etc. Another risk is, or downside, is the fact that they will, of course, be expensive, especially when the technology first emerges, which may cause even more significant class divides. Another downside is population exploitation. Now, this may seem all well and good for your parents, but imagine if a country like North Korea were to desire more desirable qualities amongst its population and to affect their genetic makeup. And then, of course, unknown unknowns, or simply put, just things we don't know of yet. In order for this to happen, it has to first be tested on humans. And, of course, since you're messing with the DNA, this will affect the lives of not just one individual throughout their lifetime, but their lineage as well. But isn't this a bit extreme? After all, you wouldn't do this, right? You wouldn't have designer babies. Well, to a degree, we already do this. After all, in the UK, 90% of known Down syndrome pregnancies are terminated in a way that is modifying based off of genetics. Now, it's not possible yet, but in China, they've recently had a successful genetically modified human embryo.
Human genome projects have had various cultural impacts over the years on various indigenous groups. We will examine various benefits, some of the negatives, and some conspiracy theories that people have been gathering because of the new technology being used and some of the unknown implications of it. So one of the benefits of various human genome projects is understanding population migrations and the cultural effects and historical clues that can be gathered through these. One of the major examples of this is archaeologists found human remains in Australia that predate bones found anywhere else in the world, and it baffled the anthropologists, scientists, because they didn't understand how people could immediately leave Africa and wind up in Australia, figuring either they sailed across the ocean or they somehow managed to leave no trace behind while walking across the land. And so, upon doing the research, they discovered that they did in fact originate out of Africa before most other populations had even exited Africa. And it's historical clues like this that can give us a better understanding of history and the origin of the human species, and can have important significance in the cultures that it affects and the populations that this kind of research can affect in a positive way. Some of the negatives, however, go along with the information side of human genome projects. One of them is the mishandling of research. For example, various researchers were looking into this indigenous group, trying to find diabetes, which had been plaguing this indigenous group for a long time. And while they did do some testing for diabetes, the primary focus of a lot of their research was on inbreeding and schizophrenia, which showed an immense amount of distrust between this indigenous group and various other indigenous groups towards this kind of human genome research. There are also political implications that can be quite damaging to indigenous groups. One of them is, say, a native group in central Brazil were they had DNA testing done on them, and it was uncovered that the population in question had, in fact, immigrated to that region only a couple hundred years ago. That would possibly give the government per se, justification to evict this indigenous group, which is something that has worried a lot of other indigenous groups in the past. And since a lot of time these places rely on oral traditions, it can be extremely worrisome to them. Now for the conspiracy theories. The main one that I found intriguing and interesting to look at concerned IBM. National Geographic Project, which is through National Geographic. One of their main backers is IBM, and IBM got a lot of their early income through providing the computer punch cards that the Nazis used during World War II for the database system on their death camps. And this conspiracy theorists theorize that since they would have no issue doing that, then they would be a-okay with developing eugenics-style bioweapons to specifically target indigenous groups or other populations. And while it does sound very far-fetched and highly unlikely, it is interesting to worry about what other kind of implications this kind of technology can have on indigenous people that could end up being extremely catastrophic if left into the wrong hands.